Hello and a very warm welcome to our online viewers. Thank you for joining today's Science AAAS webinar entitled A High-Resolution Look at the Human Cell, Introducing the Human Cell Atlas. I'm Sean Sanders, Editor of Custom Publishing at Science, and it is my honor to be the moderator for this webinar. Resolving the spatial distribution of the human proteome at a subcellular level greatly increases our understanding of human biology and disease. A high-resolution map of the human cell has been generated as part of the Human Protein Atlas database, and this provides the in situ localization of over 12,000 human proteins at a single cell level, covering 30 subcellular structures and enabling 14 major organelle proteomes to be defined. The Cell Atlas reveals that approximately half of all proteins localize to multiple compartments, and that many proteins show cell-to-cell variation in terms of protein abundance or spatial distribution. In this webinar, we will introduce our viewers to the new Human Cell Atlas and outline how it is being used to define the spatiotemporal organization of the human proteome at a subcellular level. I'm very pleased to introduce our speakers to you now for today's webinar, Dr. Mathias Uhlén and Dr. Ellen Lundberg, both from the KTH Royal Institute of Technology in Stockholm, Sweden. Thank you both so much for being on the line with us today. Before we get started, I'd like to share some information for our online viewers. At the top right of your screen, you'll find photographs of today's speakers and a view bio link which you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion, as well as a PDF of the slides. After the speaker presentations, we will have a short Q&A session, during which our speakers will address some of the questions submitted by our online viewers. So if you're joining us live, start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box, and clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Atlas Antibodies for sponsoring today's webinar. Now I would like to introduce our first speaker, Dr. Mathias Uhlén. Dr. Uhlén founded the Science for Life Laboratory in Stockholm, Sweden, where he served as director from 2010 to 2015 and has authored more than 750 publications in bioscience with a focus on the development and use of affinity reagents in biotechnology and biomedicine. He is leading the international effort to create the Human Protein Atlas with the aim of systematically mapping the entire human proteome. Dr. Uhlén is a member of the Royal Swedish Academy of Engineering Sciences, the Royal Swedish Academy of Sciences, the European Molecular Biology Organization, and the National Academy of Engineering, and is president of the European Federation of Biotechnology. A very warm welcome to you, Dr. Uhlén, and thank you so much for joining us today.
Thank you very much. I will also want to welcome everyone when they are listening. What we will do today is to go through the Human Cell Atlas, look at the new features of the Atlas, and this will be mainly by Ellen Lundberg, but I want to give you a short introduction to the Human Protein Atlas. So I will also talk about the integration of transcriptomics and proteomics data, a little bit about the TakeHome project that we started last year, and the validation of antibodies that we are very much focusing on. And then Emma Lundberg will talk about the subcellular localization of proteins, multilocalizing proteins, single cell protein variants, and then end up by the definition of organelle proteomes and the actual cell atlas. So as an introduction then, the Protein Atlas started actually 14 years ago. It is funded by a non-profit organization here in Sweden called Knut and Alice Wallenberg Foundation, and we have now spent more than 1,000 person years on this project. This is a knowledge base portal for scientists and researchers, but we also have created a resource of reagents such as antibodies, which we have passed QC that we've done, recombinant protein fragments that we are now using for target proteomics. And then lately, and I will say a little bit about that, is full-length secreted proteins that can be used for functional assays. The mapping of the spatial distribution of the proteins is our prime focus here. We do it with antibodies, DNA sequencing, mass spectrometry, and CyTOF. And in this way, we can map the different proteins in the different parts of both the organelles, the cells, tissues, and organs. And our main focus is immunohistochemistry, immunocytochemistry, transcriptomics using next-generation sequencing, and then also targeted proteomics. The way of using antibodies then gives us very good resolution. We can actually map all the proteins with single-cell resolution and actually even subcellular resolution. We can also see the distribution of the different human proteins in the context of neighboring cells. And this will, of course, provide biological and functional context to the about 20,000 proteins that we have in the human body. Up till now, we have created about 13 million images that has been looked upon by pathologists. This is the main focus has earlier been IHC, but we have also immunohistochemistry, but we've also now produced a lot of images with ICC, and you will hear more about that later in this presentation. The first map that we created was a tissue-based map of the human proteome. This was published first in Nature in 2014, and then in the Science article in about 2 years ago in January 2015. And in this article, we were then describing the tissue-enriched proteins and the housekeeping proteins and so on. And one of the highlights that we saw is this enormously focus on tissue-enriched proteins in the testes of humans, which is much more than the brain. And this, of course, caused quite a lot of attention at the time. But what we are now doing is then moving on from the 76 different cell types that we did in the first version, corresponding to 44 normal human tissues, and we are now extending this to more specialized tissues, such as the eye, part of the brain, hair, full adrenal gland, and so on. And this is now this is going on as we speak. Another effort that we're doing is also to try to do a more detailed study of the brain. And this is done in the mouse brain, where we are doing 120 different annotations of the brain to allow us to 3D reconstructions of the whole brain. And this, of course, is a good complement to the images that we have of the human brain in the normal tissue atlas. One of the things that is an advantage for us is that we are comparing then the RNA and the protein levels in these different tissues, and using a target proteomics approach that was a collaboration with Mathias in Germany. We were able then to actually look in detail on the correlation between RNA and protein levels, and in an article a few months ago, we concluded that protein levels can be estimated from RNA levels if you do it in a gene-specific manner. And this, of course, means that we can use then this genomics tool, RNA sequencing, to actually study the corresponding proteins, which is very useful. So a lot of the work in the Protein Atlas now is to integrate different types of omics data, and especially transcriptomics and proteomics data. And we now have transcriptomics data both from our own effort, the HPA, but also from the GTEx consortium in the United States and the Phantom 5 consortium in Japan. and this is now integrated into the protein atlas. And if you look into a specific gene, such as, for example, the ACE gene here, you get an expression summary, how that gene and that protein are expressed in the different parts of the human body, and actually the expression levels across all major tissues and organs are presented both on the protein level, the RNA level, using HPA, DTEX, and Phantom5 then. The take-home message from the Science article about 2 years ago then was that we were quite surprised to see about half of the genes expressed in all tissues of the human body, very few proteins which are unique for one tissue, and also that many drug targets are localized to all tissues, which is of course very interesting from a pharma perspective. The protein atlas then, we now have about 150,000 visitors every month. We are very pleased to see that a large part of this are scientists in the United States, but actually the country which is second is China, and then comes UK and Germany. So we're seeing, but we actually see visitors from more than 200 countries around the world. I just also want to mention a project that was just started, and that is also to try to produce all the 2,900 secreted proteins in humans that are estimated from the genome sequence. So we are doing this through high-throughput production in CHO cells, and in this way we can create a resource, a reagent for drug discovery and development. And this is a collaboration with the Novo Nordisk Fund and also with the company AstraZeneca. This project is advancing in a very nice way. We have a success rate of approximately 70% from design of the gene to the purified protein, and we are now trying to then produce all the human proteins that are secreted. I also want to say a few words about validation of antibodies. There has been a lot of literature lately, also warning people for the problems that on antibodies, that they are not reproducible and they are not specific and so on. So in order to try to formulate guidelines, we started an international working group, which I was chairing about 1.5 years ago, and actually we presented then some guidelines just a few months ago. One of the important things to discuss here is the pros and cons with different antibody types. We are using a lot of polyclonal antibodies in the Protein Atlas project. The advantage with that is that polyclonal antibodies show a very good functionality across applications and are not so sensitive to sample conditions for the simple fact that they have several epitopes. Monoclonal antibodies, on the other hand, that we are also using, have an advantage when it comes to batch-to-batch variations, but they are more prone to sample conditions or difficulties with sample conditions. So we're using both the polyclonal and the monoclonal antibodies in our project. The international working group that we started published a paper then in Nature Methods about in the fall, in October in 2016, where we proposed that one should use 5 pillars for validation of antibodies. And I don't want to go through that in detail, simply to say that this is our strategy now in the Protein Atlas to try to move and formally validate all our antibodies using these 5 pillars. So what we did then back in 2014 was to launch the Tissue Atlas. We then moved on and launched the Brain Atlas in 2015. In the end of 2016, we launched the Cell Atlas that you will hear about very soon. And then later this year, we hope to then launch a Pathology Atlas to actually look at the proteins which have alternative expression in cancer, and then next year move to a metabolic atlas, and finally in 2019 hopefully then release a plasma atlas in order then to describe the human protein in different tissues and different contexts in the human body. So I also, before I end, I just want to say that this resource can, of course, and knowledge base can also be used for medical problems and medical questions. And one such program we started is the S3 program here in Sweden, where we use these resources to actually go through 100 healthy volunteers that are analyzed every 3 months. And we're looking at the microbiome, the whole genome sequence, plasma profiles, blood cell status, and so on. And this is, of course, very, very interesting. So in conclusion then, I just wanted to say that we are systematically mapping the building blocks of humans in this project. All the data is open access without no restrictions. We do this mainly by spatial proteomics, so showing where are proteins in cells, tissues, and cancers. But we're also integrating transcriptomics and proteomics data. And the focus for us is the validation of antibodies, and then moving into different biochemical applications. But of course, the main focus here is that all of you can use this information free of charge without any restrictions. And with that, I want to hand the word back to Dr. Sanders, and then and Emma will talk more about the cell atlas.
Fantastic. Thank you so much, Dr. Uhlén. As we said in the beginning, our next speaker today will be Dr. Ellen Lundberg. Dr. Lundberg is currently the director of the Cell Atlas, part of the Swedish Human Protein Atlas program. She heads the cell profiling group in the Department of Proteomics and Nanobiotechnology at the KTH Royal Institute of Technology, where she is an associate professor. Her research is at the interface between bioimaging and proteomics, and aims to define the spatiotemporal organization of the human proteome at a subcellular level in an effort to understand how variations and deviations in localization can contribute to cellular function as well as disease. To achieve this goal, her lab has developed technology platforms for large-scale immunostaining and systematic validation of antibody specificity, and has initiated citizen science efforts to refine the Human Protein Atlas database. Welcome, Dr. Lundberg, and thank you very much for being on the line today.
Thank you. So I'm going to present to you the Cell Atlas that was launched in December, the end of last year. And this is work that is being done as part of the Human Protein Atlas project, and we are creating a high-resolution map of the subcellular human proteome. And compartmentalization of biological processes is a fundamental principle of living organisms, ranging from organs to organelles, and this is in order to gather the proteins needed for a specific task at the particular location. So knowing the protein localization provides clues for protein function and serves a basis for interaction studies. Defects in compartment organization also underlie the cause of many forms of human disease. So knowing the subcellular location of all human proteins would help researchers to understand protein function, cell biology, and disease. And this is what we try to enable through the Cell Atlas. We've been working on this for over 10 years, and we have established a platform, a robust platform, so that we can generate data in a systematic way. And this platform starts with cell cultivation. All proteins are localized in the U2S osteosarcoma cell line, and then we pick 2 other proteins based on a panel of cell lines, and we do this selection based on RNA-seq data. So we pick highly expressing cell lines for every protein. And then we do immunostaining using pipetting robotics, and then we do imaging, and we do high-resolution confocal microscopy. And finally, we analyze the images, we classify the patterns manually, and we also do a computational image analysis. And I will show you some examples of images. And we always use a certain set of reference markers in the images. In blue, we have the nucleus, DAPI staining. In red, we have a monoclonal antibody staining the microtubules. We also have an antibody-based marker for the endoplasmic reticulum. So these markers are present in every single image in the atlas. And on top of this, we add the HPA antibodies that stains every human protein one by one. And our goal is then to classify the pattern of this green staining. And in this case, we would conclude that this is a cytoplasmic protein. This is a mitochondrial protein. This protein is localized to the endoplasmic reticulum, the Golgi apparatus, the plasma membrane, the nucleoplast, nucleoli. And when we started the project, we classified the 10 major organelles in the cell. We quickly realized that there's so much more information in these images. There's substructures and different textures, patterns that we can see. So we have spent a lot of effort the last 5 years to classify even more patterns that I'm going to show you some examples of now. Now you can see classifications of lipid droplets, for example, the nucleoli fibrillar center, the rim of nucleoli, nuclear speckles, nuclear bodies, mitotic spindles. And this image, I think, nicely illustrates the power of using an antibody-based and imaging-based approach to study proteins, because we can see that this protein has a very distinct localization to the centromeres in the mitotic cell up in the right corner, and then at the end of mitosis it redistributes to the cytokinetic bridge. So this protein is probably not expressed at a very high level, and it's only present in the cell at certain time points, and it has a very distinct location. We can also find more dynamic structures, such as the rods and rings. It's a reversible structure formed as an adaptive metabolic response to the disruption of glutamine homeostasis. There's a handful known proteins that localize to this structure, and its function is not fully understood yet. And we have now been able to identify 18 other proteins that localize to this structure that are publicly available in the Protein Atlas. So we hope that this novel information can shed new light on the functional understanding of this intriguing structure. So altogether, we are classifying 32 different cellular structures, and we have localized over 12,000 proteins. And every protein is localized in up to 3 cell lines. And if we see that a protein is localized to multiple compartments, we classify the localization into a main and additional compartment. And as Mathias said, it's very important to be aware of potential false positive results when you work with antibodies. And we are using these pillars that Mathias described also in the Cell Atlas, or we're using 3 of them that I will show you some example of. My favorite method is the genetic one, where you use siRNA or CRISPR-Cas9 to knock out or silence the protein of interest, and you want to see that your signal disappears, as you can see in this example. That's a very solid proof that the antibody is binding the protein it's intended to target. We also use proteins that are tagged with GFP. As you can see in this image, GFP is shown in purple, and then we stain this cell with antibodies, and we want to have a good co-localization between the GFP and the antibody signal. Then we can also be fairly confident in that the antibody binds the right protein. And finally, we also use the strategy of independent antibodies very frequently. So here we have 2 antibodies that target the protein that localizes to focal adhesion sites and actin filaments, and we can see that the similar is very, very similar, if not identical. So this is also proof that the antibodies are very likely binding the right protein. But what is important to think of here is that it's important that the antibodies bind different epitopes of the target protein, so that they are truly independent. So in the atlas, we classify all our obtained locations with a score based on these validation results. So overall, we have tested many antibodies, and about half of them end up being published on the Cell Atlas. And the basic level is called approved, and this means that before an antibody can be published on the atlas, it has to pass our internal quality control procedure. The gene has to be expressed at the RNA level in the examined cell line, and the antibody has to give a selective and specific pattern on the protein array analysis. So if we have no information about the protein location, there's no literature known for this protein, but it passes our internal QC protocol, then it will be published on the atlas with the score approved. If a protein also has experimental information in the UniProt database that supports our location, it will be given the score supported. If we have validated the location with any of these 5 pillars that Mathias described, the location will be validated. And in some cases, there might be contradictory data. Maybe our location is contradicting to what is known about this protein, but we still have other proof that supports our data. Then we will publish that antibody with the score uncertain. So these scores are provided in order to help the users of the atlas to select which reliability level they want to work with. And of course, our long-term goal is to validate all antibodies and proteins in the protein atlas. And as you can see now, about 1,500 proteins are validated, and half of the proteins are approved. And this also means that all of these proteins have no experimental data about their subcellular location in the UniProt database. So here we provide novel localization data. So the Cell Atlas that was recently launched, it's an open access database, part of the Human Protein Atlas, and it's based on hundreds of thousands of images. And in this release, we've created knowledge-based chapters as entry points to help the users to explore cellular proteomes in an interactive way. And they are illustrated by the icons that you can see on this slide. And I will go through these chapters one by one to exemplify the information that you can find in them. So the first chapter is the cell line transcriptomes. And in the Cell Atlas, we have a panel of 59 human cell lines where we've determined the gene expression by RNA sequencing, and we've classified the patterns of gene expression across the cell line panel into some different categories. I think the most interesting category is the red one, the cell line enriched genes. So that's genes that are expressed more or less only in one cell line. We classify genes also as being group enriched, cell line enhanced, or expressed in all cell lines. So as you can see in the pie chart to the right, that about 2,400 proteins show a selective expression in one or a few cell lines, and about half of all genes are expressed at more or less similar level across all cell lines. And you can also have a look at this data on a gene-by-gene basis. So I'm going to show you some examples. This is a mitochondrial protein that is classified to be expressed in all cell lines. And as you can see in this chart, the level of expression is rather similar across all 60 cell lines in our panel. But if we look at another protein, such as an intermediate filament protein, nestin, then we can see that the expression is much more selective, and this gene belongs to the category cell line enhanced. So this tool is a very good tool to use if you want to look for a suitable model cell line for your experiment, or if you want to see if your protein of interest is expressed in your cell line that you're working with. So moving on to the more interesting chapters that actually deal with proteins, we have the multilocalizing chapter. I showed you a lot of images previously with nice patterns of single organelles, but in fact most images don't look like this. Many images look like this. Here we see a protein that is localized to the nucleus and the plasma membrane. And here's another example of common combination where we see localization to the mitochondria and to the nucleoli. And this is actually very common, and altogether 51% of all proteins localize to more than one compartment in the cell. And this phenomenon is common across all structures and organelles that we have analyzed. And it's of course very interesting to speculate about the biological implications of this phenomenon. This multilocalization could be have more a simple function of a spatial confinement to control the timing of the molecular function in the designated compartment. It could also be so that multilocalizing proteins have context-specific function and moonlight in different parts of the cell, and in this way increasing the functionality of the proteome and also the complexity of the cell from a systems perspective. So the multilocalizing proteins is something that we will put more research effort into in the future. Chapter 3 describes the single cell variations, and some images looks like this, where you can see that the protein of interest, shown in green, is only expressed in some cells. There's a clear on and off expression here. Sometimes it's only a few cells that express the protein of interest, and sometimes we see more of a gradual variation. In this image you can see that the cells with a large nuclei express the protein at a higher level than the cells with a smaller nuclei that express the protein at a lower level. In some images we can also see multilocalizing proteins where only one of the compartments vary. Here we can see the protein in the nucleus in all cells, whereas the cytoplasmic localization only is present in some cells. We can also see more distinct spatial variations. This might be hard to see, so you have to look closely. You can see that there's one cell that is expressing the protein in the cytoplasm at a high level, but then if you look closely, you can see that the protein is actually present in the centrosome in all other cells in this image too. And sometimes we see complete translocations of a protein. Here we can see a protein localized to the Golgi apparatus in all cells, except in the two cells that have just divided, where the protein is localized to the endoplasmic reticulum. So even though these are static images, this kind of variation between the cells in the image indicates that there is a dynamic behavior of the expression of this protein. And there can be many reasons for this dynamic behavior, but one plausible reason is that it could be correlated to cell cycle progression, since these are non-synchronized log phase growing cells. And in total, we see this kind of single cell variations for 16% of all human proteins. And in contrast to the multilocalizing proteins, this phenomenon is more prominent in some cellular compartments. It's mainly seen in the nucleus, cytosol, nucleoli, and mitochondria. And we have also determined that half of these variations are correlated to the cell cycle. And what the remaining half is caused by needs more research to figure out. And in order to investigate the cell cycle correlation in more detail, we have created a computational model to infer the cell cycle position from the Doppler microtubule reference markers available in all images. And I will give you one example. This is a protein KIF23 from the atlas, and you can see that the green intensity varies between the cells. So then we can apply this computational model to do a pseudo-temporal analysis of the protein expression, and we can model it over the cell cycle progression. And chapter 4 relates to the organelle proteome. So we've localized over 12,000 proteins to 32 cellular structures. We've done a lot of functional enrichment analysis and interaction analysis and so on. But in order to package this information for the users of the Cell Atlas, we have created and defined 14 major organelle proteomes as outlined here. This is a screen dump from the Atlas. And I'm just going to show you an example of what it looks like by showing you the mitochondria. So for every organelle, we start by a brief introduction describing the organelle, and we show some examples of well-known organelle proteins. We also describe the structure of the mitochondria, and in the top table here, we recommend 6 different proteins and antibodies that can be used as markers for mitochondria and different substructures in the mitochondria. The second table shows the 10 most highly expressed mitochondrial proteins based on our RNA sequencing data. And then we also show examples of different cell lines and how the morphology of the mitochondria can vary between cell lines. We also, if there is an organelle with a lot of different substructures, we also show examples of the different substructures. Then we describe the function of the organelle in question, and we show the results of enrichment analysis that we've done using the gene ontology terms, and we can see that the enriched terms for the mitochondrial proteins are all related to the known functions of the mitochondria. We also go more in depth into the multilocalizing proteins. So here you can see a cytoscape plot where the mitochondrial proteins that are shared with other organelles are illustrated, and the blue colored dots are combinations that are overrepresented, significantly overrepresented compared to the background of the whole proteome. So we can see that mitochondrial proteins are often also seen in the nucleoplast and in the nucleoli. And this plot is interactive. If you click on any circle here, you will get to a list of these proteins, and it will easily take you into the detailed protein pages on the atlas. And finally, we also investigate the gene expression patterns of the mitochondrial proteins across all the tissue samples present in the protein atlas. And here we can conclude that mitochondrial proteins are significantly more likely to be expressed in all tissue types compared to the background of all human genes. So this is the kind of information that you can find in these knowledge-based chapters. You can use as entry points and educational material in the Protein Atlas. So the Cell Atlas, this is the starting page of the Cell Atlas, can be found at proteinatlas.org. It's an open access database based on hundreds of thousands of images. And we have these knowledge-based chapters. But you can also, of course, search for your protein of interest, any gene in this atlas, and then you can just type it into the text box below here. And I will show you an example of cyclin B1 just to illustrate the data that we have in the atlas and what it looks like. So when you search for a protein, this is the page that we will get to, and here we have a schematic cell that illustrates where we have localized the protein, and the top gray box also shows some general information about this gene and protein. The second box shows a summary of all our information. And the third box shows a summary of the data reliability. In this case, we have validated this antibody, this localization based on 3 antibodies and siRNA and GFP staining. And at the bottom of the page, you see something called assays. And this is all the different assays that we have done for this gene. And if you click on it, you will get to that part and those results in detail. So I will show you all of them, starting with the RNA expression data. Again, we can see that cyclin B1 is expressed in all cell lines. You can also sort this table in different ways. The next section is the main part where we see the images, the localization images. You can compare 3 images side by side. You can compare the different antibodies, same antibody. You can toggle the marker channels on and off, and you can also turn on an intensity lookup table if you want to compare the intensity in different neighboring cells. You can also turn on the object to see the segmented cells. You can also click on the image to see the high resolution image in our virtual microscope, and also here you can toggle the channels on and off. The next section is something that we call custom data. So this can be different data from gene to gene, protein to protein. Sometimes it's a super resolution image using STED microscopy. Sometimes it's colocalization with a particular cellular marker. And in this case, we have stained the Fuji cell line to demonstrate the cell cycle dependency of cyclin B1 expression. And then you get to the validation data. We have knocked down cyclin B1 or silenced cyclin B1 using siRNA, and we can see that the intensity of the staining disappears, and we've quantified this. And we've also used cells expressing cyclin B1 tagged with GFP, and we can see that we have a good co-localization for the staining. And we have also localized the protein in mouse cells. So to get back to the starting page, in the very top you have a summary of all results, but if you're interested in looking at the raw data by yourself, go down to the respective section and you have all of this information available. So to conclude and summarize the Cell Atlas, it's a freely available map of the human subcellular proteome, and knowledge about the protein subcellular location provides clues for protein function. And spatial information is a very important complement to quantitative RNA protein metabolite measurements for systems biology. And for the future, we aim to provide a final, complete, and validated Cell Atlas. And we aim to validate using these 5 pillars that Mathias described, validate every single antibody in the atlas. We also want to quantify the plasticity of the spatial subcellular distribution of the human proteome and understand how it contributes to cell type specificity as well as disease. And we want to generate a holistic whole proteome subcellular model of a canonical human cell over the course of one cell cycle. Then I have 2 more small things I want to mention. So when we launched the Cell Atlas, we also had a poster in Science magazine. So if you want a copy of this to put in your lab, you can order it at the link at the bottom of this page. And then finally, I also want to mention the citizen science projects and thank the 200,000 gamers all over the world that has contributed to the Cell Atlas project. So in order to reclassify a lot of these patterns that we have refined in the Atlas, we have taken part of a citizen science effort. And we, in a collaboration with CCP Games and MMOS, we created Project Discovery, a scientific minigame in EVE Online, a science fiction massively multiplayer online game. It was launched exactly 1 year ago and it's still running, so you can still play it and contribute to the Cell Atlas. But up till now, over 200,000 players have contributed with 18 million image classifications, and they have spent over 60 working years doing so in total. So please try it out and help us refine the details in the Atlas even further. And with that, I would like to end and thank the Cell Profiling Group and the whole Human Protein Atlas team and all our collaborators that help us to make this very useful atlas. And I also want to thank our funders, the Ellen Lundberg Foundation, the Royal Institute of Technology, Ellen Persson Foundation, SciLife Lab, and Atlas Antibodies for sponsoring this workshop. Thank you.
Fantastic. Thank you so much, Dr. Lundberg, and thank you to both of our speakers for their wonderful presentations and for sharing the Cell Atlas with us. We're going to move right on to some questions submitted by our online viewers. A quick reminder to those watching live that you can still submit your questions by clicking the Ask a Question button below the slide window, typing the question into the message box, and clicking OK. I've also had a number of viewers ask about getting a copy of the slides. You can find a PDF copy of the slides by going to the Resources tab below the slide window and clicking on the Event Slides link, and that should download a copy for you. So let's start off the Q&A portion of the webinar with a question for you, Dr. Uhlén. So what makes the Protein Atlas and the Cell Atlas special, and what separates it from other efforts to do similar types of mapping?
So thank you for that question. So what we are doing, I think it is a rather unique approach to actually probe and spatially localize all the proteins using antibodies, but also to integrate it with other types of technologies, including proteomics and transcriptomics. There has been lately announcements of other types of Cell Atlas projects. For example, the BioHub in California, and also the Cell Atlas project in Seattle, funded by the Allen Foundation. These are similar efforts, but they are also very complementary to what we are doing. Our focus then is to do this kind of holistic genome-wide, protein-wide analysis using antibodies that we have generated ourselves, but we also have more than 50 commercial companies that has provided antibodies to our effort, and we are very grateful for that.
Fantastic. So let me give the next question to you, Dr. Lundberg. Are you developing or planning to develop a protein atlas of mutated proteins with mutation-specific antibodies?
No, we're not planning to generate anything like that at the moment. We are not generating any mutation-specific antibodies right now, but if you do have any mutation-specific antibodies, we'll be happy to try them. I believe that it's more likely that we will move into perturbations rather than mutations at this point. But it's a very interesting question.
Great. I had a second similar question asking about whether you have antibodies that distinguish any splice variants.
Yes. So we have some antibodies that do. So we try to make 3 to 4 different antibodies for every target protein, and we try to design them to target all isoforms, but sometimes we also make antibodies that target 1 or 2 different isoforms. So actually in the protein atlas, you can see the exact antigen sequence that was used. So you can see if the antibody is targeting 1 or 2 or 3 and how many of the isoforms it's targeting. So all of this is publicly available, and you find it under the antigen tab in the atlas.
Okay, the next question, I think maybe we'll stay with you, Dr. Lundberg, for this one. Why is RNA data important for determining the subcellular localization of proteins?
Yes, so it's not, when we started the project, we didn't have RNA sequencing data. So it's not important for localizing the protein per se, but if any cell line expresses about 10,000 to 14,000 proteins. So if we know, it's important to help us to pick the right cell lines to localize the protein in. But that's more or less what it's relevant for.
Great. Dr. Uhlén, back to you for a sort of a general question. Do you have a sense of how many proteins are in the protein atlas relative to the total the total number of proteins that are found in a human cell?
So if we talk about the protein coding genes, there is about, what we expect now from the Genome Project, we have about 19,500 protein coded genes. We now have about 16,000 of those we have right now, or actually 17,000 right now. So we are way over 80% of them now covered. But obviously, as the question you asked before, there is also the splice variants, the isoforms, the different types of modifications, and these are of course also very important for understanding cell biology. But what we're trying to provide then is a map of the sort of basic proteome with one antibody covering every protein-coded genes. So in the cell atlas we have about 12,000. So we are expecting then that to increase, and hopefully then in a few years we will have a full coverage of all the human proteins.
And perhaps you could answer this question as well. I thought this was interesting. The viewer says they know the the cell atlas was released in December, but for how many years prior to release have you been actively producing data and images for the cell atlas?
Yes, so we took the first image in 2006. And we started scaling up slowly. So the first couple of years has been about scaling up the processes, making robust platforms for large-scale immunostainings. And then that would be the first 3 to 4 years. And then we spent 3 years just generating a massive amount of data, and then we spent the last couple of years to still generate data, but also to refine the details. So in total, we've been going on for 10 years.
Great. Another question for you, Dr. Lundberg. How do you control for the fact that cells in culture might have different expression levels or localization of their proteins compared to native human cells in vivo?
Yes, so that's something that is that we are very interested in, and that's why we're also expanding our cell line panel with more primary cells, immortalized cells, and also stem cells to get more normal types. And we've been trying to compare our localizations with the localization of the protein in the tissue atlas and also the cancer atlas. And of course, we don't have the same subcellular resolution, but we can compare roughly nuclear cytoplasm at least. And in general, I would say give and take around the majority of the proteins localized to the same compartments, but there are some differences, and we do see a higher degree of nuclear localization of proteins, both in the cell lines, the subcellular atlas, and in the cancer images too. So whether this is due to cross-reactivity of the antibodies or if it's due to that the protein is actually falsely localized, we don't know yet.
Okay. Dr. Uhlén, you talked about antibody validation in your presentation. Have all of the antibodies used in the cell atlas been validated? And if so, how is this shown?
So we have an antibody validation page where we show exactly how we have validated the antibodies, and we're also very cautious to also show the primary data so any researcher can go in there and actually see how the antibody has validated. And just as Emma was describing, we then base our validation or score them in 4 different categories, everything from the highest validation, which is the formal validation using the pillars that was suggested by the working group, and then all the way down to some antibodies that we actually call uncertain. These are not that many antibodies, and we still want to show them because we think that they might be correct, but we also want to show the researcher that these should probably be, we should validate them and get help to get them validated by the community or by more work. So, but in most cases, the antibodies have been quality controlled using our standard procedures, and they are there to everyone to see in the antibody validation pages.
Dr. Lundberg, this viewer says, I don't see any siRNA data for my protein in the cell atlas. Does this mean that the antibody has not yet been tested, or was it not successful?
It means that the antibody has not been tested. So if we do a validation experiment and would find out that the antibody is not binding the intended target protein, we would immediately take it off the atlas. So it means that it's not tested.
Great, thank you. And I believe this question was answered, but I wanted to just make sure that the audience knows. This viewer asked if it's how would it be possible and under what conditions, for instance, price and subscriptions, would they be able to get access to the Human Cell Atlas? But as far as I know, it is completely free for all users. Is that correct?
Yes, everything is completely free for all users. You don't even have to register to use the atlas. It's freely available, and you can also download all data. There's only one thing that is not available at this point, but we're working to make it available, and that is the high uncompressed images. So right now it's only compressed JPEG images that are available in the atlas, but we hope that we can make the whole data set of uncompressed TIFF images also available later this year.
Okay, fantastic, great. So the next question is, this viewer says, I missed the source of the antibodies. Are they commercial or are they generated within the project?
So I can maybe answer that. So most of the antibodies have been generated in-house, but what you can, but there are also about I guess about 10,000 antibodies that have been provided by other resources. We have made sure that all the antibodies used in the protein atlas are commercially available, so they can be used by scientists around the world. And the easiest way to see what antibody and where they are from is to go into the antibody information page, where there is a link to that antibody and the antibody provider that you can get access to it. So we have actually made sure that we are only using antibodies available for researchers around the world.
Great. So the next question, possibly Dr. Lundberg, this one would be for you. The viewer asks how protein expression is quantified and follows up with asking, can low levels of protein expression be reliably detected and reported, for instance, transcription factors? So perhaps you could share what the sort of the low-end detection level is for the proteins in the cell atlas.
Yes. So the signal intensity of which is what we measure from the antibodies, it depends on many different things, and it depends on the affinity of the antibodies and how many epitopes you're recognizing and so on. So we can't really say that, compare two different antibodies and say that this protein is more strongly expressed than this protein. So we can't compare it protein to protein, but we can compare within one image. And what we can also say is that the signal in our images is not dependent on the total concentration of the protein, but rather of the local concentration of the protein. So sometimes we can get super bright signals of, for example, a centrosomal protein or protein localized to nuclear bodies, even though the actual copy number of that protein is very low, but we get a very high, they're localized to a very distinct part of the cell, and then we get a high signal. Whereas if we would lyse the cells and run mass spectrometry or lyse the cells and run a Western blot, we might not be able to detect that protein because we disrupt this high local concentration. So I would say that we can detect, it's a very good method to detect low abundant proteins as long as they are specifically localized.
And following on from that, fixation of samples especially before immunocytochemistry, immunohistochemistry, can affect the localization and intensity. Is this important? Is something that you look at when doing these experiments?
Yes, we have a publication on that, or several publications on that. So we look a lot on that, and we know that the fixation and permeabilization, of course, affects your results. So we're using a rather standardized PFA fixation, and we permeabilize the cells with Triton as our first round experiment. And we know, for example, that we do get a bit more cytoplasmic and nuclear background by doing so. And if this protocol doesn't succeed, we proceed with a methanol-based protocol. But we do know that, for example, if you use methanol to fixate and permeabilize your cell, you will lose a lot of the soluble proteins. So that's why we always start with a cross-linking protocol.
So we are almost out of time for the webinar, but I wanted to put one more question to you both, and let's start with Dr. Uhlén. Just what is the future of the protein atlas, and maybe more specifically the protein cell atlas? Where are you going next? What can we expect to see in the future?
So the major new thing coming is the pathology atlas that we're releasing this summer. We have used a lot of data from the Cancer Genome Atlas and combined that with our own data, and we are hoping then to launch a completely new pathology atlas, which shows what proteins are expressed in the different cancers, but also then compare that with the clinical outcome of the patients in these cancers. So we're very excited about that. And then we will release a metabolic atlas showing which enzymes are expressed in the different tissues of the human body, and metabolic models, what metabolites are then produced in the different parts of the body. And we will do that in collaboration with people at Chalmers in Sweden, and hope to launch that in the spring of 2018. And then the next major launch is the Plasma Atlas, where we want to look at the levels of different proteins in human plasma, both healthy individuals but also diseased individuals. But at the same time, we also want to keep on working with the quality and the validation of the antibodies and make both the cell atlas, the tissue atlas, and the pathology atlas more complete. So it will then provide what we think an excellent resource for life science scientists around the world.
Fantastic. Well, that is, I think, a perfect way to end the webinar. So it just remains for me to thank our speakers, Dr. Mathias Uhlén and Dr. Ellen Lundberg from KTH Royal Institute of Technology. Please go to the URL now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation within approximately 48 hours from now, and I can guarantee that these slides in PDF form will be available together with that on-demand version. We're interested to know what you thought of the webinar. Please send us an email at the address now up in your slide viewer, webinar@AAAS.org. Again, thank you so much to our panel and to Atlas Antibodies for their kind sponsorship of today's educational seminar. Goodbye.